will enjoy your presentation.
Thank you, Andrea. Let me share my screen. Yeah. Okay, so good afternoon, everybody. Can you all see my screen and hear me well? Okay, great, thanks. So firstly, I would like to thank Andrea for inviting me to speak here today at the CGIBD Research Seminar Series. As an associate member of the center, I'm very excited to have this opportunity to interact with everybody here through the Zoom today. As Andrea just mentioned, my lab has been working on 31 and its related physiology in the past many years. So in my talk today, I'm going to give you some updates about our recent research, including some unpublished results. So any suggestions and comments will be highly appreciated. So my lab is interested in metabolism, which is a set of very complicated chemical reactions that occur in all living organisms in order to maintain life. Metabolism has a number of essential functions. However, what interests us the most is the observation that many intermediate metabolites, such as acetyl-CoA, SAM, alpha-ketone glutarate, and NAD, can function as a co-substrate or cofactors for a number of modification enzymes that can modify DNA, histones, many other protein factors, and even RNA. So all these modifications can dramatically impact every step of the central dogma in response to environmental changes. So metabolism is a key mediator of environmental influence on physiology. So in my lab, we are interested in understanding how these intermediate metabolites interact with protein modification enzyme to regulate cellular function. To do this, we have been focused on a very unique protein modification enzyme called 31. So 31 belongs to a family of highly conserved NAD-dependent protein deacetylases known as sirtuins. So as you can see here, this family of proteins are able to use NAD as a co-substrate to remove the acetyl group from the protein substrate. Recent studies have shown that many other acyl groups with different lengths and different features of the side chain can also modify the lysine residues on the protein through a similar mechanism. And the sirtuin family member are able to remove these different types of acyl modification with different specificity, but in an NAD-dependent manner. So this NAD-dependent activity makes sirtuins a very unique family of metabolic sensor that can integrate the metabolic signals from two important intermediate metabolites, acetyl-CoA and NAD, with protein modification, epigenetics, gene expression, as well as cellular energetic status. So they are important for cellular response to the environment. In my lab, we are focused on the regulation and the function of 31, which is the most conserved member of this family in mammals. 31 is predominantly a nuclear protein, and its protein substrate including transcription factor, cofactors, splicing factor, and histones. So today I plan to divide my talk into two parts. So in the first part, I will focus on 31 and its related metabolism in mediating host microbiome interaction. And in the second part, I plan to share with you an unpublished result on diet-gut microbiota interaction in regulation of anti-tumor immunity. So this project was originated, started from the 31, but now have nothing to do with 31. So I know that I'm going to have a lot to talk about that. I will try to make my story simple, but if I confuse you, you have any questions, just feel free to interrupt me during my talk. So let's talk about 31. 31 is a highly dynamic protein. So its expression and activity can be regulated by a number of environmental factors. For instance, we and others have shown that 31 can be activated by phosphorylation in response to environmental stress, including nutritional stress induced by high-fat diet feeding, chemically induced genotoxic stress, as well as heat shock induced by high environmental temperature. However, since SIRT1's activity is exclusively dependent on NAD, so cellular level of NAD is one of the major regulator of SIRT1's activity. So one of the major focus of my lab is to study environmental regulation of cellular NAD metabolism. So NAD is a nicotinamide adenine dinucleotide. It is best known for its function in the central energy metabolism, where it accepts electron from the nutrient molecules through NAD-dependent dehydrogenase to produce NADH, which then gives these electrons away to oxygen through the electron transport chain, generating a membrane potential that is required for ATP biosynthesis. So NAD is important for ATP biosynthesis. In mammals, there are three major ways to synthesize NAD. The first pathway is the salvage pathway from nicotinamide. And the nicotinamide in the cell can be either uptaken from environment as a major component of vitamin B3, or can be released from NAD by a number of cellular enzymes, including sirtuin, as I just showed you. So the rate-limiting step of the salvage pathway is the conversion of nicotinamide into nicotinamide mononucleotide, which is catalyzed by an enzyme called NAMPT. The second pathway starts from nicotinic acid, known as press-Hunter pathway. Mammals can also synthesize NAD from the essential amino acid tryptophan, involve a much longer pathway. The second and the third pathway are converged at the nicotinic acid mononucleotide, then convert to NAD and NAD. Since the major differences between nicotinamide and nicotinic acid is the amine group in the nicotinamide, so this salvage pathway from nicotinamide is also known as amidated pathway, where this press-Hunter pathway is known as deaminated pathway. So in mammals, these two pathways are separated because we don't have an enzyme to connect nicotinamide with nicotinic acid. Because of essential function of NAD in the cell, so disruption of NAD homeostasis has been associated with a number of human diseases. For example, many cancer cells have increased cellular level of NAD in order to meet their high energy demanding. As a result, several of the chemical inhibitor targeting NAMPT rate-limiting enzymes in the salvage pathway have been developed for potential cancer treatment. On the other hand, it has been shown that NAD level decreased during aging, and this reduction is believed to be associated with a number of age-related diseases, particularly for neurodegenerative diseases. Therefore, a number of NAD boosting supplements, I believe many of you must have heard about that, NR and NMN, have been actively promoted by the nutraceutical industry as anti-aging supplements. So one thing you may notice already is that all these manipulations are focused on the salvage pathway. This is because a paradigm in the field is that this salvage pathway is the main cellular source of NAD in mammal, whereas the press-Hunter pathway is just a minor pathway, is not important. However, recently, we discovered that bacteria, including mycoplasma contaminated in the tissue culture cells, as well as commensal bacteria in the gut, can link these two pathways together in mammal. And this bacteria-mediated deaminated NAD biosynthesis contributes substantially to tissue NAD biosynthesis, especially after supplementation of precursors from this amidated pathway. So the story started when Igor Schatz, the staff scientist in my lab, accidentally found that cells contaminated with mycoplasma are protected from NAMPT inhibitor-induced death. As you can see here, when human cells are cultured in the media containing about 8 micromolar of nicotinamide, they are hypersensitive to the STF treatment, which is NAMPT inhibitor. However, when they are contaminated with mycoplasma, they are protected from this death. So this protection also occurs in vivo. As you can see here, when xenografted into the tumor, into the node mouse, this tumor contaminated with mycoplasma are protected from NAMPT inhibitor-induced inhibition of proliferation. So how could this happen? So working together with John Liu in Jason Locase's lab at Duke in a metabolomic analysis, Eager figured out that mycoplasma can in fact dramatically increase cellular abundance of NAR and NAD. If you remember correctly, what I just showed you, those two metabolites are two important metabolites in the DM-related NAD biosynthesis pathway. So mycoplasma can also dramatically increase the concentration of nicotinic acid and nicotinic acid ribocytes in the medium, in the culture medium. So all these observations suggest that mycoplasma could protect cells from non-PT inhibitor-induced cell death by enabling this alternative DM-related NAD biosynthesis. Indeed, in this stable isotope tracing experiment using deuterium-labeled NAM, that's nicotinamide, Eager shows that when cultures are clean with no bacterial contamination, all the NAM are converted to NAD through NMM. So if you treat the cell with STF, then cellular NAD biosynthesis is completely blocked and the cell die. However, if cells are contaminated with mycoplasma or E. coli, in this case here, we can see that the NAD is converted, NAM is converted to NAD through both pathways. Then treatment of STF is not able to deplete the cellular NAD level and the cell survived. So what is going on there? So we were able to figure it out. Many microbial, but not vertebrates, have an enzyme called PNCA that can convert NAM into nicotinic acid, nicotinamide into nicotinic acid. And we show that this PNCA-mediated conversion of NAM into NA is not only necessary, but also sufficient to protect the cell from non-PT inhibitor-induced cell death. For instance, in this cell survival assay, wild-type E. coli is able to protect human cells from STF-induced cell death, just like what I showed you. But deletion of the PNCA gene in the E. coli completely abolished this protection. We, in fact, don't really need bacteria to be here. We only need this enzyme. So overexpress this E. coli PNCA genes in mammalian cells, protect them from STF-induced death. We actually don't even need the bacteria. We just need its products. Bacteria, I mean, this enzyme, we only need its products. So supplementation of nicotinic acid in the culture medium protects the mammalian cell from STF-induced cell death. So this result here is not really exciting. This has been known for years. But our point here is this nicotinic acid comes from bacteria. So we also show that this PNCA-mediated DM-related NAD biosynthesis is also important for NAD synthesis in vivo. So in this experiment, we repopulated the germ-free mice with either wild-type E. coli or PNCA knockout E. coli. We then garrotted them with NAM and followed the tissue NAD metabolites 3 hours later. You can see here, in mice repopulated with wild-type E. coli, we can see labeled NAD metabolites in both M-related and DM-related pathway. You can also see here. However, in mice repopulated with PNCA knockout E. coli, and all the labeling in the DM-related pathway is completely gone. So I would like to bring your attention that on the final total NAD levels in tissues. You can see that in colon, particularly in the liver, mice repopulated with PNCA knockout E. coli have dramatically reduced level of newly synthesized NAD, which is a heavy labeled NAD here, indicating that this PNCA-mediated DM-related NAD biosynthesis contribute greatly, largely to tissue NAD biosynthesis after supplementation of the NAM. So therefore, in contrast to what was previously believed, so in mammals, these 2 branches of NAD biosynthesis pathway are also linked due to the presence of microbial PNCA activities. So then what is the physiological significance of this connection? So using a triple isotope labeled NAM, we were able to show that when taken orally, NAM was converted by microbial PNCA to NA, started in the small intestine, but largely in the colon. This is not surprising because colon have a much higher bacterial load than the small intestine. So this PNC-mediated conversion contribute to more than 30% of NAD synthesized in the small intestine, but more than 60% of NAD synthesized in the colon. So then many intermediate metabolites in this deaminated NAD biosynthesis pathway in the intestine, including NAMN, NA, and NAR, can then be transported to the portal blood, contributing to more than 80% of NAD synthesized in the liver and more than 60% of NAD synthesized in the kidney. So you might notice that this deaminated NAD synthesis appears to be more efficient than the amidated NAD biosynthesis, despite the fact that the concentration of NAN in the portal blood is about several hundred-fold higher than that of the deaminated precursor. So this is because the rate limiting enzyme in the salvage pathway, the NAN-PT, is gated by NAD, meaning that NAD can feedback inhibit its activity. While the rate limiting enzyme in the deaminated pathway, the NAPRT, does not have this feedback inhibition mechanism. So in addition to NAM, we also showed that NR, which is one of the most popular NAD boosting supplements, when taken orally, can also be degraded by bacteria and then contribute to the NAD biosynthesis through this deaminated pathway. So our study have a number of important implications. Firstly, I think our study can maybe helpful to explain the failure of NAN-PT inhibitor in the clinical trial, and suggest that a combination of NAN-PT inhibitor together with the inhibitor against the microbial PNC enzyme may be a better strategy for cancer treatment. Our study also suggests that the dysregulation of gut microbiota might underline age or some disease-associated decline of NAD, and suggests that manipulation of gut microbiota together with the NAD boosting supplements may be a better strategy to increase NAD synthesis in these disease conditions. So therefore, our study indicated that, in addition to the environmental factor that I just showed you previously, microbial can also potentially regulate SIRT1's activity through NAD. So then what about the function of SIRT1? So in the past almost two decades now, we and others have shown that SIRT1 is a master regulator of metabolism, stress response, inflammation, and development by targeting different type of important transcription factors and cofactors in different tissues. And one of our most favorite system, of course, is the gut. I think for the audience here, I don't need to convince you the importance of the gut, also how does the gut look like. So here I just want to simply show you that SIRT1 is widely expressed in all intestinal epithelial cells, including enterocytes, intestinal stem cells, and Pannett cell, which is the type of innate immune cells originated from intestinal epithelium. So in order to study SIRT1's function in intestine, we generated an intestinal-specific SIRT1 knockout mouse model by breeding SIRT1 flaxed allele with winning queen mice. So these mice have deletion of SIRT1 in all the intestinal epithelial cells. The SIRT1 eye knockout mouse are typically normal under normal feeding condition when they are young. So the only defect we can see is in their enterohepatic circulation of bile acid. So this is a process in which bile acid synthesized in the liver are released to the duodenum after feeding to help to absorb lipid, and then are transported back to the liver through portal circulation at the distal ileum. So in each cycle, there is about 5% of loss of the bile acid from the feces. The liver will synthesize the actual bile acid in order to maintain the constant level of total bile acid pool. So what we found is when we delete SIRT1 in the intestinal epithelium, this lead to reduced immune absorption of bile acid. So then this lead to increased loss of bile acid from the feces and reduced circulation level of bile acid in the blood. Also, liver now synthesizes more bile acid in order to compensate of this increased loss. So when these mice are challenged with diet containing high bile acid, then they have reduced accumulation of bile acid in the liver due to this reduced immune absorption of diet and bile acid. So they are protected from bile acid-induced liver damage. So then what is the mechanism? So we figured out that 31 actually is able to regulate bile acid metabolism through transcriptional regulation of FXR, which is a master regulator of bile acid metabolism, as well as ASBT, which is epic bile acid transporter. So we found that this impact of 31 is at an upstream transcription factor called HNF1 alpha, as well as its dimerization factor called DCOH2. So what we found is DCOH2 is actually acetylated protein and is the substrate of 31. So in control mice, 31 is able to deacetylate DCOH2 and increase its ability to dimerize with HNF1 alpha and increase their binding to the promoter of FXR and SPT. So this action of 31 promote linear bile acid transport. In 31 inoculum mouse, on the other hand, the DCOH2 is hyperacetylated. Then they prefer to form a heterotetramer instead of homotetramers, homotetramer instead of heterotetramer, which is HNF1 alpha. So then this H1 alpha dimerization is reduced, and they have reduced binding to the promoter of this important genes involved in bile acid metabolism, which reduce immune bile acid transport. So interestingly, when this mouse getting older, we start to see additional phenotypes in addition to bile acid metabolism. For example, at the molecular level, this intestinal 31 knockout mouse, they start to have increased NF-kappa B signaling, but defective mitochondrial metabolism and have increased oxidative stress. So then at the morphological level, starting from 6 to 8 months old, we start to see hyperactivation of their Paneth cell as this is stand by Paneth cell marker lysosome. And also around about 1 year old, we can see increased lymphocytes infiltration in their colon, and the mouse actually are sensitive to DSS-induced colitis. So another thing we noticed that is in the aged 31 inoculum mice, they are depleted a special class of gram-positive bacteria bacillus. So from here, this 16 fecal bacteria 16S rRNA gene sequence result, which is done by Andreas Kohl here. So we found that aged 31 inoculum mouse have reduced abundance of 5 genera of bacteria in their feces. And all 5 of them belongs to the Bacillus families, including the most abundant Lactobacillus. As many of you know, that is one of the major components in the probiotics available in the market. So through a number of studies that I don't have time to show you today, so we were able to figure out that this Diet-microbe, this depletion of bacillus in the age 31 knockout mice is due to their increased bile acid loss in their feces. Because compared to many gram-negative bacteria, these gram-positive bacillus families are hypersensitive to bile acid. We were also able to show that if we eliminated these Diet-microbe differences, then the inflammation phenotypes of 31 knockout mice are also gone. However, the hyperactivation of Paneth cell phenotypes seems to be independent of gut microbiota. So currently, we generated a Paneth cell specific 31 knockout mouse model using DAFA6 Cree, and we are characterizing the phenotypes of these mice. So to summarize what I have showed you in the first part of my talk, so I hope I have convinced you that metabolism play a major role in regulation of host microbiome interaction. So what I showed you is biota is able to interact with our diet and then modulate NAD metabolism, which could potentially regulate 31's activity. I also showed you that 31 could also regulate gut microbiota through regulation of bile acid. So then in the second part of my talk, I'm going to switch gears and talk about methionine metabolism. So I think methionine, everybody knows that, is an essential amino acid required for protein synthesis. So methionine actually have many unique features. So one of most striking features is methionine is one of the 4 sulfur-containing amino acids in our body, and it is important for synthesis of SAM, which is a major methyl donor for methylation, including DNA methylation, histone methylation, and RNA methylation. Methionine is also important for production of hydrogen sulfide, which is a gas signaling molecules recently have been shown to be important for angiogenesis and also for aging and stress response. Another important function of methionine is to synthesize glutathione, which is important for the maintenance of redox homeostasis. So what makes methionine metabolism even more unique is the observation that dietary methionine restriction have been shown in many studies in many model organisms that can extend the lifespan and the delay aging, which is similar to what caloric restriction is doing. So another feature is dietary methionine restriction can also prevent metabolic diseases, including obesity, insulin resistant, and diabetes. So methionine also restriction have also been shown that can control cancer cell proliferation. So in many preclinical model and xenograft model, dietary methionine restriction has been shown to suppress tumor growth and also can inhibit the tumor initiation capacity of the tumor initiation cells and also sensitize cancer cells to chemotherapy and radiotherapy. So the reason why we get interested in methionine metabolism is about several years ago, we found that in mouse embryonic stem cells and embryos, 31 is able to regulate methionine metabolism and then the sensitivity of these cells and embryo to methionine restrictions. So we hypothesized that since the yes cell and the cancer cell are metabolically very similar, so we hypothesized that 31 might also regulate methionine metabolism and also the sensitivity to methionine restriction of the cancer cell. Of course, the result turned to be negative, which is not surprising. So we found that in the liver cancer cell, it is not a 31, but is another important transcription factor, HNF4 alpha, dedicates their sensitivity to methionine restriction, both in vivo and also in xenograft model. So then we were wondering whether HNF4 can really determine the sensitivity of tumor to methionine restriction in vivo. What I mean in vivo is not in xenograft model, is really in vivo using a genetic cancer model. So to test this hypothesis, we used a HNF4 alpha positive cancer model, that is APC min mice. I think many of them must have know that APC min mice is a genetically mutated mice, have a mutation on the APC genes. So this mutation leads to the defective degradation of beta-catenin, therefore induce expression of a number of oncogenic genes. So the heterozygous mice develop spontaneous intestinal adenoma, and also this adenomas are sensitive to nutritional manipulation. So high-fat diet can enhance their formation. So we, to test our hypothesis, we decided to feed APC min mice with either a control diet containing 0.8% of methionine with no cysteine, or methionine restricted diet containing 0.17% of methionine, again with no cysteine. So based on our studies from previous xenograft experiment, as well as numerous literature reports, we expected that this HNF4 positive APC-mean tumors are going to be sensitive to methionine restriction. They are going to have reduced formation of tumor upon methionine restriction. However, Min Ji, the postdoc who is leading this project, is surprised to find out that mice fed with methionine restricted diet actually have increased tumor number in their small intestine, as well as increased tumor burden in their colon. So what is most striking is that APC mice have some clinical phenotype starting when they became about 4 months old. So they are going to have rectal bleeding, you are going to have increased body weight, and have a number of other clinical signs. So we were required to remove these animals from our study based on our animal protocol. So on the regular and controlled diet conditions, this symptom-free survival of the APC mean mice have a median lifespan about 180 days, that is about 6 months. However, methionine restriction dramatically shortened their mean lifespan to about 4 months. So then when we first see this result, the first mind come across our head is there must be something wrong with our control diet. Okay, so although this control diet is a specially formulated defined diet to control this methionine restriction diet, so there may be some unknown components in this diet that somehow suppress the tumor formation. So then to control this situation, we decided to use another pair of the methionine diet, that is our control chow diet that contain 0.4% of methionine, or a methionine supplemented diet containing 1.3% of methionine. So this is a custom-made diet. So we know that the only differences between these two diets are the methionine we added to this diet. So again, as you can see here, for mouse fed in this diet, animal fed with a diet containing lower methionine diet have an increased tumor number in the small intestine and an increased colon tumor burden in the colon, and have reduced lifespan. So then what is going on there? So we were really actually puzzled by this result, because this is really completely against what our own experiment, xenographic experiment, also against many other reports that is documented in the literature. So then after scratching our head for a long time, we realized that there is a major differences between our study and the study that previously conducted either using xenograph model or preclinical model. That is, in the previous xenograph model and the preclinical model, immunocompromised mice are used. Okay, so for example, node mice. However, the APC mice or APC mean mice, they are immunocompetent. So it has been shown that methionine actually is important for immune cell, especially for T cell activation and differentiation. So this is actually not really surprising, because given the important function of methionine in protein synthesis, also in metabolism. So then we were wondering, maybe the difference we saw in our APC mean mice is due to their different host immune system status. So to test this, we decided to use a synergetic tumor model using mouse colon cancer cells, CT26 Y-type cells. So this is the cells that can be allografted into an immunocompetent BABC mouse and form tumor. And since we are very naive to immunology, so we decided to use NSG mice, which is one of the most immunocompromised mice that lack B cell, T cell, and NK cells as a control. So as you can see here, you can appreciate that in the immunodeficient NSG mice, methionine restriction can suppress tumor growth, just like what we saw before in the xenograph model and also the literature reports. However, methionine restriction completely lost this effect when the tumors are in the immunocompetent BBC mouse. So when this experiment was repeated using the 1.3% diet and 0.4% diet, so you can see even more dramatic differences. So in the immunodeficient NSG mice, methionine restriction repressed tumor formation. However, in the immunocompetent NSG mice, methionine restriction promotes tumor formation. So therefore, as we suspected, so the tumor's response to methionine restriction is really dependent on the status of host immune system. Consistent with this observation, when tested for the response of this tumor to immuno checkpoint inhibitor, we found that the allograft CT26 CL25 cells, which is a subclone of CT26 wild-type cell that have a better response to immunotherapy, they are sensitive to anti-PD-1 antibody when the mice are fed with control diet, whereas they are resistant to this treatment when mice were fed with methionine restricted diet. Also consistent with our suspicion that methionine restriction can impair anti-tumor immunity. So when in both APC min mice and also in BBC mice, methionine restriction can reduce the circulating CD8 positive T cells. Also in the BBC mice, we are able to detect the increased exhausted T cell, which is TIM3 positive, and also immune suppressed Treg cell. So then our question is, how does methionine restriction impair anti-tumor immunity? So as you know, this actually is not a really novel question because there are a number of studies have shown that methionine have cell autonomous impact on T cell activation. One of the studies as I was just showed you. So it also one of the most popular mechanism is methionine restriction can reduce cellular level of SAM, which is a major cellular methyl donor. And this reduction of SAM reduced histone 3 K4 trimethylation on some key genes involved in T cell growth or cytokine production. Also on the histone 3 K79 trimethylation on STAT5 promoter. STAT5 is key modulator of T cell activity. So then, but you know, since we have a microbiota in mind, so we were very happy to see that it had been reported that gut microbiota can interact with the diet to dramatically regulate immunity. So then we hypothesized that diet methionine may modulate anti-tumor immunity, at least in part through modulation of gut microbiota. To test our hypothesis, we first analyzed whether methionine restriction can really alter bacterial composition in animal. The answer is yes. Again, Andreas Kohl did this sequence for us. So this impact actually is pretty major. So as you can look at it in the FINIA level, methionine restriction can reduce the bacterial datis and enhance firmicutes. So therefore, can significantly increase the ratio of these two major phenom of bacteria. Also, in almost all tested animals, methionine restriction almost completely wipe out another major phenom of the bacteria, that is vertical microbial. So when we look at that more detailed level, we can see that methionine restriction can dramatically reduce this A-mucin phenom and also reduce odor factor, but increase the abundance of bifidobacteria. So since the changes is pretty major and pretty dramatic, so we designed a fecal transplantation experiment in which we fed a tumor-free Black 6 donor mice with either control diet or methionine restricted diet, and then transplant their fecal bacteria into antibiotics treated recipients APC min mice, which are fed on the chow diet. So we then analyze the tumor formation in the recipient mice 3 to 4 weeks later. So it's really remarkable. So this 1 week of the fecal transplantation is sufficient to induce the tumor formation in the APC mice from methionine restricted donor animal. Also increase the tumor number and the tumor burden. So when we look at the small intestinal CD45 party immune cell population in this donor mice and also fecal recipient mice by single cell RNA-seq, we can see that based on their individual gene expression and marker gene expression, so this CD45 party of immune cell in the small intestine can be clustered to about 7 functional groups. And then from here, you can see that methionine restriction in the donor animal, methionine restriction alone is sufficient to reduce the CD8 party of effector cell, as well as CD8 central memory T cells, but unexpectedly expand the B cell population. But strikingly, so this reduction of CD8 effector cell, CD8 central memory cell, and expansion of the B cell can be transferred to the recipient mice simply for after 7 days of the fecal transplantation. So indicating that diet-methionine indeed primarily impact the gut immunity through regulation of gut microbiota. So then our next question is, how does the gut microbiota impact the T cell function? So we decided to focus on the metabolites because that is a direct product from dietary intervention. So we did a metabolomic analysis of fecal metabolites, again, in collaboration with Jason Snipe at Duke. And he was able to find a number of changes in donor mice, but none of these really changes are really preserved after about 3 weeks of fecal transplantation. So the only defect we saw is a fecal hydrogen sulfur production activity. So as you can see here, diet-methionine restriction significantly reduces the fecal hydrogen production activity in the gut microbiota in the donor mice, and this reduction was preserved after about 3 weeks of transplantation into feces. So then we suspect that maybe hydrogen sulfide have something to do with the T cell activation, consistent with what our thoughts. So it has been, hydrogen sulfide is a gas signal which has been shown to be important to a number of important functions in the T cell, and it has been shown to be a T cell potentiator. So to test our hypothesis, we decided to supplement mice under methionine transplanted with fecals from methionine donors with GYY4137, which is a water-soluble donor of hydrogen sulfide. As you can see here, this supplementation is sufficient, is able to rescue the increased tumor numbers in the small intestine and dramatically rescue of their health span. So then we also test the supplementation of these hydrogen sulfide donors in methionine restriction-induced resistant to anti-PD-1 antibody in the immunocompetent mice. As you can see here, for this CT26 CL25 cells, as I showed you before, they are sensitive to anti-PD-1 antibody when the mice are fed in a control diet, but they are resistant to these antibodies when mice were fed with methionine diet. However, supplementation of hydrogen sulfides is sufficient to rescue their resistance to this therapy. Again, as a control, none of this effect occurs in the immunodeficient NSG mice. So indicating that all this impact of diet-methionine restriction as well as hydrogen sulfides impact on tumor growth is completely dependent on the host immune system. So here is a working model to summarize what we know so far. So we think that in the microenvironment in the intestine, so there is a very complicated interaction between nutrient, cancer cells, and immune cells. And then as an essential amino acid important for protein synthesis. So methionine actually are required for both immune cells and the cancer cell. So the net effect of methionine restriction on tumor growth in these conditions is really dependent on the relative dependence of cancer cell and immune cell to this nutrient. So we show that in our study, gut microbiota play a additional layer of regulation on the sensitivity of the cancers to methionine restriction. So we show that in addition to this cell autonomous requirement by the host cell, methionine can be help to boost the microbial production of hydrogen sulfide. And this hydrogen sulfide can indirectly activate anti-tumor activity. And our study also showed that this activation, this hydrogen sulfide production from the gut, also has some systemic impact on anti-tumor immunity. So then to summarize what I showed you so far, so as I said that my lab is interested in studying how metabolism interact with protein modification enzyme to regulate cellular function. And then our study indicated that two important environment factors, diet and microbiota, can interact with each other and then dramatically impact this interaction. So finally, I would like to thank people who did the work. So I kind of mentioned it during my talk. So this microbial regulation of NAD metabolism was spearheaded by my staff scientist, Igor Schatz. And then my methionine restriction and anti-tumor project is led by a postdoc fellow Ming Ji with assistance from my biologist, Qin Xu. And the 31 and biased metabolism project was led by a former postdoc fellow, Lev Zad, and Monique and Anicias worked on the age-dependent effect of intestinal 31 on intestinal inflammation and the gut microbiota. And finally, I want to say big thanks to the to a UNC CGIBD community. So as you know, I was trained as a biochemist, and I come with a background that really primarily focused on mechanistic study on 31. So when I first decided to work on the gut about 10 years ago, I didn't know anything about the gut. So then I have my all three former postdocs trained in person, trained by people from Susan Hannes lab, from Keenan's lab. Also, Agile helped us a lot in all this in our 31 eye knockout mouse project. And we also have a project studying the role of 31 in intestinal tumor genesis. And we get a tremendous help from Bob and Top to get the clinic sample. And also currently we are working with Quinn Lu, for this methionine restriction project, and hoping using his platform to identify additional metabolites that may be responsible for dietary methionine's impact on anti-tumor immunity. Also, as I also mentioned that all of our gut microbiota sequence project are done by Address Core. And also we have a collaboration with Notibiotic centers to make some germ-free animal in our model to study the interaction between 31 and the gut microbiota. And finally, I would like to thank Yi Song from the Department of Microbiology and the Immunology Department. So because we are very naive in immunology, and he really helped us to look at all our fact sorting results, make sure that we don't make any naive mistakes. So I want to say thanks to everybody in here. And I think without your support, we are not able to really follow our data and question for our research. So with that, I will stop here. And if you have any questions, feel free to let me know.
Thank you so much. I think we have time maybe for a couple of questions. I see Balfour. Are you raising your hand or you're saying goodbye?
Well, actually, I'm clapping as well as, so that was fantastic. That was really, far-reaching and lots of information. I could ask questions for an hour, but on your first part, your SIRT1 knockout mouse had inflammation. And my first question is whether that was in the colon versus small intestine or both.
Oh, yes, for our 31 intestine knockout mouse model, right?
Yes.
Oh, yes. The primary phenotype we observed is in the colon. And okay. For the small intestine, we didn't really look at very carefully. It could be there as well. But we know that the bile-acid defect is really in the small intestine.
Yeah, well, the reason I asked was because of, what do you think is the mechanism? I mean, obviously, metabolic defects in the intestine, particularly the high metabolic rates in the goblet cells and the Paneth cells could be very important. But you showed the Paneth cell defects, but that small intestine obviously can spill over to the large intestine. What do you think is the mechanism of that inflammation? It's probably multifactorial.
Yeah, it is multifactorial. So actually I probably didn't explain that part very well because, I want to show you another stuff, okay? So basically, yes. So basically, yes, we see a number of defects in this animal model, especially when they get older, okay? So as I said, there's a lot of the defect either at the molecular level. They have a number of the defects at the morphological level. So what we were able to figure it out that, for the gut microbiota defect, this is because of bile-acid. So remember, I showed you that when we delete 31 in the small intestine, they have problem to reabsorb the bile-acid because their effects are signaling pathway is defect. Okay, so this end up have a high bile-acid in their colon because they cannot absorb it, and all this bacteria all this bile-acid now get lost from the colon. So this increased concentration of bile-acid actually kill bacteria because bile-acid is a very potent fatty acid to kill bacteria. Okay, and then we have done a number of very detailed characterization. So we show that because of this bacillus, they are gram positive, and it has been shown that their member structure is different from gram-negative bacteria, so they are more sensitive to bile acid. Okay, so this is a major reason for the gut microbiota defect. And the inflammation, we believe most of them come from the bacterial defect because we did some experiment to eliminate this bile acid defect. So once we eliminate this bacterial defect, the defect in gut microbiota is gone and the defect in inflammation is gone. Okay. However, the Paneth cell, this activation is independent of this because even after we did we done all this, we get rid of difference in the gut microbiota by antibiotic treatment, or we treat the cell with bile acid sequester to eliminate the difference with bile acid. This hyperactivation are still there. Okay, so then that's the reason why we made this 31P knockout. This is Paneth cell specific knockout mouse model. So that is what we are doing in the germ-free mice with your core. Okay, so in this model, they still have Paneth cell hyperactivation. So we think 31 have an intrinsic ability to promote Paneth cell proliferation, but we're still working on that.
Yeah, well, my suggestion is work with Xiaoling Li to look more over than total bile acids because some are protective, some are aggressive in the gut, and particularly primary, secondary, conjugated, unconjugated. See what the differences are in the colon. And I also would suggest you look at goblet cells because the Paneth cell, if the nutritional aspects are with the SIRT1 is messed up, I bet his goblet cell function might also be abnormal.
Yeah, and, that's a great suggestion. Yes, we do see some goblet cell abnormality as well in our mouse model. So we are still trying to figure out the mechanism, but your suggestion is very good one. Okay, thanks.
I have a question. Well, I don't know if anybody else has questions. I have a question about the you know, in the first part of your talk, you showed that you had sort of the host mechanism for generation of NAD, and on the left you had that the bacteria could contribute NAD with via the tryptophan pathway or by the metabolism of tryptophan and nicotinic acid. So my question is, when you have, so you have that inhibitor that inhibits the
NAMPT.
Yes, this pathway. But what happened when you look at the other pathway, at the bacterial pathway, in the context of tumor cells and in the context of aging and both? So the inhibitor plus the bacteria in different contexts.
That's a good question. So that we are doing that right now. The reason why there are very, if you check in literature, there are very few studies focus on this DM-related pathway. The reason I just mentioned to you, because people believe this part is more important, and this part is just minor. So one very interesting thing is when we send our paper out to review, and one reviewer criticized the importance of our study, because the comment is, it is well known that this pathway is not important at all for mammal. So why you guys are doing this? So then, so we were saying this exactly the reason why we are doing this, because we found this pathway is important. Okay, so their study right before our paper comes out, showing that actually this pathway, as you mentioned, it's very important actually for cancer. So this study is published in Nature in 2019, late 2019 or early 2020. So what it shows that, as I mentioned to you, many cancer cells have increased NAMPT. That is the expression at transcription level. But their study shows that many other type of cancer cells, they have the gene duplication of gene involved in this pathway. So they are also hyperactive, but the mechanism is different. It's not at the transcription level, it's at gene copying level. And also they showed that in that study, if you inhibit this pathway by inhibitor, and you can really actually help to prevent, to limit tumor growth. Yeah, it is important. And now we are making some animal model, try to study more about this pathway mechanistically. So that is ongoing.
Great. Again, this was a really, really exciting talk, and I'm very happy that you were able to tell us the story. So if there aren't any other questions, I would like to thank everybody for coming, and thank you so much, Xiaoling, for sharing your research.
Thank you. Thank you for your invitation. It's my pleasure.
Bye, everyone.
Bye.